بسم الله الرحمن الرحيم والصلاة والسلام على سيدنا محمد وعلى آله وصحبه أجمعين. Assalamu alaikum wa rahmatullahi wa barakatuh, dear students. I hope you're all well, you and your families are all safe at home, and I hope that you have all settled well into this emergency distance learning. So let me start first by introducing myself. This is Dr. Behja Al-Riyami, one of the instructors of this course, and today, inshallah, I'm gonna take over from Dr. Lamia, which I'm sure that you've all enjoyed the previous lectures and you have learned a lot, whether it was a continuation of Unit 2 or the beginning of Unit 3. So today we are going to talk about cell membranes. Basically, every single cell has a cell membrane, whether it was bacteria, plant cell, fungi, protista, animal cells, they all do have a cell membrane. So we're going to study the cell membranes in details. We're going to see the structure in details and what does every single component does in that membrane. Then we're going to move into the outside of the cell membrane. So these are surfaces. The surfaces represent the cell walls in plants and the extracellular matrix in animal cells. After that, we're going to talk about junctions. So junctions are basically the point where two cells come together. So it's the joining of two cells. Junctions معناتها تقاطع. All right. Let's go ahead. All right. So what do we expect by the end of this lecture? What are the important things that we expect from you by the end of this lecture, inshallah? All right. The first thing, we are going to learn that biological membranes are arranged in what we call a fluid mosaic model. All right, so this sentence might seem scary, but it's not. You'll see in a bit and you'll understand it in details. So we'll talk about fluid. Why do we call it fluid? Why do we call it mosaic? All right. Then the second thing that we're going to talk about are the components of cell membranes and their functions. So what are the different components of the cell membrane? They're made up of what exactly? And what does each component do in that cell membrane? And the third learning outcome is you are going to recognize the structure and function of surfaces. And these are the cell walls in plant cells and what we call the extracellular matrix. Extracellular, the word extracellular means that it's present outside of the cell membrane. It's on the exterior of the cell membrane. All right. And the fourth learning outcome for today is we are going to identify the different intercellular junctions. Intercellular means it's between one cell and another. Okay, and junction, as I mentioned in the first slide, it's the cross between the two cells. It's how they are joined together, what's there in between. And we're gonna discuss 3 cellular junctions that are present in animal cells and 1 that is present in plant cells. All right, so before we go and deeper into Unit 3.4, let's refresh our minds with a summary of Unit 3. All right, if you recall that with Dr. Lamia, you've learned all about the internal organelles of the cell. So in 3.1, we started the endomembrane system, and you learned about the nucleus and the endoplasmic reticulum. Then in Unit 3.2, we continued with the endomembrane system, and we learned all about the Golgi apparatus, lysosomes, vacuoles, vesicles. And that was the end of the endomembrane system. Then we went ahead and we talked about the two very important organelles, which look very similar, and they have similarity in their structure, and they have a resemblance in their functions. And these were the chloroplasts and the mitochondria. Then in Unit 3.3, which was the last lecture, we learned all about the cytoskeleton. We basically talked about the three components of the cytoskeleton: the macrotubules, the intermediate filaments, and the microfilaments. These are very important. If you recall, they give the cell its shape, and they also offer support and function in movement. So this is done, this is done, and this is done. In today's lecture, we are going to learn about the boundary of the cell, because now we are done with whatever were inside the cell. So now we are actually going to the outer area of the cell. We're moving externally, and we're gonna learn about the cell membrane. As I mentioned, we're gonna learn about surfaces, and we're gonna learn about junctions. All right? And finally, the last thing we are going to learn about in Unit 3 is the transport of substances into and out of the cell. So from outside the cell to the inside, and vice versa. All right. So cell membranes are also called biological membranes. Anything that is biological, it means that it's present where? It's present inside a living organism. So we call it biological membrane. We can call it plasma membrane or cell membrane. The main function of the cell membrane is it forms a boundary around the cell. Okay? And also it controls traffic into and out of the cell. So it controls what goes into the cell and what comes out of the cell. And this traffic, we are going to learn more about it in the next lecture, Unit 3.5. All right, so what are membranes made up of? They are made up of macromolecules. Can someone tell me what are the macromolecules which we studied in Unit 2? All right, I'll let that link to Unit 2 be done by you, inshallah. And membranes are arranged according to what we call the fluid mosaic model. So let's dig deeper into this term, fluid mosaic model. Okay, so let's look at the first term, fluid. Whenever you hear the word fluid, you know that it implies movement. And this is true about cell membranes. It's not solid, you know. Maybe it gives us the impression that it's solid, but it's not solid. It's not static, but it's floating around. It's dynamic. This is the meaning of fluid. And what about mosaic? What's the meaning of mosaic? Mosaic. All right. So mosaic is when you have small pieces and you arrange these small pieces together in order to form what? In order to form a large piece of something. موزايك معناتها فسيفساء إذا سمعتوا عنها. فسيفساء. So that's the meaning of mosaic. So basically you've got many small pieces, you arrange them together to have a final big picture. And if you look into this beautiful photo of a plasma membrane, you can see that there are many things. There are many things in the plasma membrane. You've got this thing here in purple, you've got this thing in green, you've got the phospholipids, you've got this thing here. So basically you have many small pieces that are put together, and that's why we call it mosaic. Okay, now these purple things, let me highlight it here. These purple things which you see here, these are proteins. These proteins can be either embedded. Embedded means they are inserted, like this one, this one, this and this and this. Or they can be attached to the double layer, like, you choose a different color, like this one here. This is attached, it's not embedded, and this is attached. This one is also attached. We are going to talk in details about the proteins later on. Okay. Right, let me move forward. Okay, so now in the next few slides, we are going to talk about the fluid characteristics of the membrane, the fluid characteristics of the membrane. All right, so as we agreed in the previous slides, membranes are not static sheets of molecules. They are not static. Okay, the opposite of static is dynamic. They are flexible. Just imagine oil, okay, because the membrane is made up of phospholipid, so it's lipid. So imagine it as a kind of oil, okay, not that fluidy, but again, it's fluid, it's not static. Now let's look at why is it not static? Why is it moving? Why is it fluidy? So the first thing we are going to focus on is the movement of phospholipids, because phospholipid is the major component of cell membranes. Now, if we pay attention here, okay, these two phospholipids, this one and this one, they can actually drift laterally. So this is, oops, okay, this is quite dark. This is called lateral movement. Lateral movement means on the same side, from left to right or from right to left. So basically this one here can come this side and the other one can go to the opposite side. So they are basically drifting laterally in the plane of the membrane. All right. Now, if you focus on the time, this is very, very frequent. This is a frequent movement. Let me select a lighter color. This is a frequent movement. يعني it happens frequently. It happens very, very often. And actually, if you notice the number here, this is 10 to the power of 7 times per second. That's crazy. You can't even imagine this number. It means it's 10 million times per second. So that's how frequent it is. All right, so that's the first movement, which is lateral movement. Now, if we look at the second movement is called flip-flop. Flip-flop. Flip-flop means this phospholipid goes to the other layer. So they exchange from one layer to the next layer. Now it's clear here. Below, you can see the flip-flop. This one, phospholipids, it was below and now it's going above. And the other one was above and now it's going below. That's why we call it a flip-flop movement. Now, notice this movement is very rare. It doesn't happen very often like this one, the lateral movement. Why is that? Can you tell me why is this movement rare? Now, if you see here, it actually says it's once per month. So once every 30 days. It means it's really, really rare. Why? Why do you think so? Let me give you a few seconds to think about the answer. All right, I'm sure by now you know the answer. It's related to the nature of the phospholipid. Okay, it's related to the nature of the phospholipid. Now let me draw one phospholipid here. This is called the phosphate head. And these are called the what? The fatty acid tails. Now, the phosphate head is hydrophilic. This is hydrophilic, right? So it loves water. What about the fatty acid tails here? Yes, they are hydrophobic. They hate water. They want to keep away from water. Now, if it's going to do this flip-flop movement, it means that this hydrophilic head is going to get into this hydrophobic region, and the tails are going to be close to the water here and there, which they really hate water, so they can't be touching or facing water. And that's why this movement is very rare. However, on the other side, the lateral movement, you are not changing the hydrophilicity or the hydrophobicity. They are just moving laterally. So you're not messing up with what they like and what they dislike. All right, so this is about the movement of phospholipids in the cell membrane. All right, let's continue with the fluid property of membranes. As you have seen in the beautiful image of the plasma membrane in the beginning, we said that we have the phospholipids and we have the membrane proteins. All right, now let's look at the membrane proteins. Are they moving or are they static, which means they stay in one place? Are they mobile? Mobile means they are moving. Or they are stationary. Stationary means they are not moving. They are static. Static, stationary, they mean the same thing. Mobile, dynamic, moving, they mean the same thing. All right, so here it says that membrane proteins can be either mobile or they can be static. Basically, some proteins, they move in very directed manner. If you recall, we have motor proteins attached to the cytoskeleton. So maybe some of the proteins can be moved in a very guided direction by the motor proteins. While other proteins, they never move. So these are stationary. They stay in one place and they never move. So basically, we have two types of proteins. Those that move and those that excuse me, do not move. All right, let's have a look at this experiment, which shows the evidence that some proteins are drifting. Some membrane proteins are drifting or moving. Basically, drifting means that they are moving or they are carried away very slowly. All right, let me choose another color here. Purple. All right, so we have a mouse cell. Okay, and the membrane proteins in the mouse cell are represented. The membrane proteins are represented by this pink color. Okay, the pink color, they represent the membrane proteins or the proteins that are present in the cell membrane of the mouse cells. While the human cell, again, they have membrane proteins, but in this case, they are represented by a purple color just for simplicity so that you understand what's going on. All right. Okay. So the researchers, what they have done is they have fused a mouse cell with a human cell. Okay. They fused them together. So they joined them together. So now you have this hybrid cell. Hybrid cell. Hybrid cell, it means it's a combination of a human cell and a mouse cell. It's basically a combination of two different cells. Hybrid يعني هجين. Okay, after less than 1 hour, this is what we got. This is what we got. You see the result of this experiment. Okay, so you can see that the membrane proteins of the two species, the mouse and the human, are completely intermingled together in the membrane of the hybrid cell. All right, you can see the purple and the pink membrane proteins, they are all distributed around that hybrid cells. So this gives us an indication that the membrane proteins are also moving. They are also moving in the cell membrane, okay? They are not static, but they are moving, and this contributes to the fluid quality of the membranes. Of course, we already said that we also have some which are stationary. Some proteins are stationary. They don't move, and others, they do move. So we call them mobile or dynamic. All right. Okay. Now, we are still talking about the fluid quality of membranes. First, we discussed phospholipids, and second, we discussed membrane proteins, and now we are discussing more specifically about the fatty acids in the phospholipids. Okay, the fatty acids in the phospholipids. Okay, now the fluidity of the cell membrane is actually affected by the nature of the fatty acid. Can you recall the concept of oil and butter in the this was in the lipids lecture in unit 2? Why the oil is liquidy or fluidy while the butter is solid at room temperature? Please link whatever you learn in this slide to the slide or to the concept of solid liquid oil and butter that you have already learned about in unit 2. All right, let's look at this. So We have two kinds of fatty acids. We have unsaturated fatty acids or hydrocarbons, and we have saturated hydrocarbons. So, unsaturated hydrocarbons are those. Can you tell me what is an unsaturated hydrocarbons since we already covered this? I'm sure you know the answer. Unsaturated means that there is a carbon-carbon double bond. There is a carbon double bond carbon. This is the meaning of unsaturated. Okay? There is a double bond. Now, this double bond, whenever you have a double bond, you have a bending. You have bending of that hydrocarbon. All right? So if I'm going to draw a fatty acid here, if I draw it like this, there is a bend. It means that this one here, you have a carbon-carbon double bond. That's why it's bended. There is a bend or there is a kink. All right. Now, if you have an unsaturated fatty acid, so you have a kink. A kink means bend. It's equal to bend. All right. Now, if you have a bend in the hydrocarbon, it means you have got some spaces. Look at this here. There is space. There is a space over here. There is a space. And over here and here and here, etc. So whenever you have a space or gap between molecules, this is going to make the membrane less compact and therefore it's gonna make it more fluid. So let me write it here. Gaps, okay, means less compact. They are not stuck together. Okay, there is space between them. There is a gap between them, which means it has more fluidity. So it's more fluid. Okay, it's more fluid. Let's look at the other situation when you have saturated hydrocarbon, when you don't have a carbon-carbon double bond. So you have carbon here, you have another carbon. Each one has single bonds. That's why it's called saturated. Saturated. All right, when this is the situation, then you have your phosphate head here and you have your hydrophobic tails here, and we draw them as straight. They have a straight shape. Now, when they have a straight shape, this makes them pack together. So this is one, this is another one, and this is the third one. You can see how they are compacted together, which makes the membrane less fluid because there is no space. See here, there is no space. They are just joined together, which makes them more compact, which makes them less fluid. When it's less fluid, we call this viscous. Viscous. Viscous معناته لزج. All right? Okay, so you can see here how the fatty acids play an important role in the fluidity of the cell membrane. Now, let me ask you a question. Which membrane, number one, let me write number one here, and number two here. Which membrane you think is better? The one with many unsaturated fatty acids, which is number one, or the one with many saturated fatty acid, which is 2. Is it 1 or is it 2? Think about the answer, or you can Google it later and let me know which one is better in your opinion. Okay, now again we continue with the fluid quality of the membrane. We're still talking about the fluid quality of the membrane because this is very, very important and many things play a role in the, let me choose the highlighter here. Many things play a role, as you have already seen, in the fluidity of the plasma membrane. Okay. Now we're going to talk about cholesterol. Cholesterol. So cholesterol is an important component of the plasma membrane. Can you recall the structure of the cholesterol? This is cholesterol. This is cholesterol. Can you recall the structure? Actually, this figure gives you an indication of the structure. It's made up of what? 4 fused rings. This is the structure of cholesterol. So cholesterol is the second lipid component in an animal cell. They are present in animal cells but not in plant cell membranes. Okay? All right, so what does cholesterol do in the cell membrane? Let's have a look here. So the role of cholesterol is different according to the temperature. According to the temperature, the role of cholesterol differs. So at warm temperature, when the temperature is a bit warm, what will it do? It will actually restrain. Restrain means it will limit. It will limit the movement of phospholipids. It will make them move less. When it makes them move less, it means it will reduce fluidity. Why does it do that? Because at warm temperature, there is already a lot of movement. Okay, so it needs to balance. At warm temperature, these guys here, these guys, okay, the phospholipids, they are already becoming further, or the fluidity of them, they can move better, so they need something to keep them closer together. So cholesterol is going to do that. It's gonna limit their movement if the temperature is warm. Okay, however, if the temperature is cool, what it's going to do, it's going to maintain fluidity. Okay, imagine that at cool temperature, what's going to happen to the phospholipids? They are going to get closer together. But now, since you have cholesterol here, it means you have space. See, can you see these spaces here? So it will maintain fluidity. It will keep them, or try to maintain, to keep them more fluid because they are getting packed together. So you can see how the role of cholesterol differs according to the temperature. Actually, the cholesterol, we actually call it the, let me write it here, cholesterol. Okay, let me change the color. Cholesterol is called the buffer of the membrane. Do you remember what is a buffer? Do you remember what is a buffer? A buffer is something that will resist changes. It will resist changes. So when the temperature is high, okay, it's gonna do what? It's gonna reduce fluidity. Fluidity. Can you see this opposite relationship, inverse relationship? When the temperature is low, it's gonna do what? Yes, it's gonna increase, or it's gonna maintain the fluidity. acts just like a buffer. All right? Okay. So now we are done with the fluid character of the membrane. We talked about it in details. We started with the phospholipids, then we moved to the proteins, membrane proteins. Then we talked about the fatty acids, whether it's saturated or unsaturated. And finally, we talked about the role of cholesterol in maintaining fluidity of the membrane. Okay, so now we move to the second term of the fluid mosaic model. So now we're going to talk about the term mosaic. Let me choose a highlighter. Mosaic quality of the membrane. As we mentioned previously, mosaic means it has a lot of small different components. They are made up of macromolecules. The main building blocks of cell membranes are lipids. These are the main constituents of the plasma membrane. So you have got the phospholipids. That's the first lipid component, and it's the main component. And the second lipid component is what? What is the second lipid component? We just mentioned it. One slide back. It is the cholesterol. Cholesterol is the second lipid component. Okay? All right. In addition to that, we also have carbohydrates. Notice these green structures here. These are the carbohydrates. These are the carbohydrates, and they are often found in the exterior of the plasma membrane. Exterior of the plasma membrane, and you can see these are the minor components. And we also have proteins. These are membrane proteins. Membrane proteins. All right, so these are the components of the cell membrane. Okay, so now in the next few slides, we are going to talk about each component. Okay, we are going to start with the membrane lipids. The membrane lipids. Okay, so phospholipids are the main components or the major components of the or the main building blocks of the plasma membrane. Okay, we already talked about the structure before. You can go back to the unit 2.4 about lipids if you want to refresh your memory about lipids. Okay, so lipids, they have hydrophilic head. Can you tell me why the head is hydrophilic? Why the head is hydrophilic? Why does it love water? Yes, because it's made up of phosphate, and phosphate is charged. Okay, and it also has a hydrophobic tail. Can you now tell me why the tails are hydrophobic? Why the tails are hydrophobic? Because they are made up of fatty acids. And fatty acids are long hydrocarbon chains, which makes them very hydrophobic. So basically, phospholipids are made up of things that are hydrophilic and things that are hydrophobic, which means we call the molecules amphipathic or amphiphilic. Amphipathic means they are both hydrophilic and hydrophobic. All right. The second lipid component of the cell membrane is the cholesterol. cholesterol. Now, cholesterol is found only in animal cell membranes. They are not found in plant cell membranes. They are found in animal cell membranes, and they are very, very important in maintaining membrane fluidity. As we just mentioned, the cholesterol, they are called the buffers of the membrane. They resist changes in fluidity. Okay, they maintain the fluidity of the cell membrane. All right, let's move ahead. Okay, the second components are the membrane carbohydrates. Membrane carbohydrates. All right, so membrane carbohydrates are called oligosaccharides. Oligosaccharides. What are oligosaccharides? Okay, now let's go back to the lecture of carbohydrates. Okay, let's refresh our memory. Okay, now if you have one sugar unit, I can't see my pen here. Okay, it's disappearing. Let me choose a different color. Okay, if you have one sugar unit, then this is called monosaccharide, right? If you have two sugar units, then that's gonna be called disaccharide. If you have many sugar units, then it's gonna be called what? Yes, that's right. Polysaccharide. Polysaccharide. But if you have got polymers, which is polysaccharide, that have less than 15 sugar units, then we are going to call it oligosaccharide. Oligosaccharide, okay? Less than 15 sugar units. Okay, so actually this is a new term. Okay, so we start, or the simplest one is monosaccharide, followed by disaccharide, followed by oligosaccharide, and then you have the big polymer, which we call it polysaccharide, more than 15 sugar units. Okay, so let's look at the carbohydrates in this figure. Okay, as you notice, طبعا this is the interior surface of the membrane, and this is the exterior surface of the membrane. This here is the cytoplasm. If you notice, the carbohydrates are always found on the exterior. They are found on the exterior of the plasma membrane. Okay, now we have two types of oligosaccharides. If the oligosaccharide is bound to lipids, like this one here, you can see this is the oligosaccharide. It's binding to the phosphate head here, which is the lipid. Then we call it a glycolipid. Glycolipid. Glyco means carbohydrate or sugar, and lipid is lipid. So that's sugar bounded to lipid. We call it glycolipid. However, if you have an oligosaccharide like this one that is not bound to the lipid, but it's bound to what? It's bound to the membrane protein, then we are going to call this glycoprotein, which is sugar that is bounded to a protein. So we have glycolipids, which is sugar covalently bonded to a lipid, or we can have glycoproteins, which is sugar that is covalently bonded to protein. Which one is the more common? The glycoprotein is found more frequently. It's more common. It's more common. Okay, now these carbohydrates are really, really important, as you're gonna see in the next. Carbohydrates that are present in the membrane play an essential and a key role in cell-to-cell recognition. What does that mean? Well, it means the carbohydrate gives the cell the ability to differentiate between one kind of cell from another kind of cell. Let's say, for example, that we have a cell here, and we've got another cell, and a third cell, and a fourth cell. Now, these carbohydrates, which are located on the membrane, let's say this has a green carbohydrate, and this has a green carbohydrate, which act as a tag. They act as tags for the cell. And let's say this has blue carbohydrate tag, and the last one has an orange tag. All right, so the ability of this specific cell to recognize a cell, a neighboring cell of the same tag, is through the presence of this carbohydrate. And this will make it recognize that this is not from the same type of cell, and this is not from the same type of cell. This is the meaning of cell-to-cell recognition. Why is this important? Well, there are a couple of points that are important in cell-to-cell recognition. Firstly, it's important in cell sorting and organization, and this is essential during development of tissues and organs, so that, for example, the cells of the heart, they come together, and the cells of the kidney come together, and the cells of the different systems come together. This is called cell sorting and organization. The second point, or the second importance, is by the cells of the immune system. The carbohydrates that are present on the membrane gives them the ability to reject cells that are foreign from the body. For example, bacteria or viruses. Okay, so they recognize that these cells are foreign to our body, so let's reject it. Also, in terms of organ donation, if the cells do not match, then the body is going to reject that donated organ. And a very important example is the red blood cells grouping. As we all know, we have 4 red blood cells grouping: blood group A, B, AB, and O. And this will be explained in details in the next slide. Okay, now let's look at this red blood cells grouping. Red blood cells grouping. Okay, now let me ask you a question. What determines the blood groups of individuals? Why do we have different blood groups? We have O blood group, we have A blood group, B, and AB. So why do we have these blood groups? Okay, the answer to this question is, as you have seen in the title, it's due to the presence of membrane glycolipids. Membrane glycolipids. So these are carbohydrates that are bonded to the lipids components of the cell membrane. Okay, let's have a look at the O type first. Okay, طبعا this is the lipid. You can see it's a glycolipid, so this is the lipid part, and this is the carbohydrate or the sugar part. Okay, you can see this is the sugar found in the membrane of the red blood cells, that is O type. Now, if we look at the A type, it's exactly the same as the O, but what's the difference? Can you see this? Let me change the color. Can you see this blue thing here, which is not present here? So that's the difference between the A and the O. Just one tiny sugar. One tiny sugar. It's called N-acetylgalactosamine. N-acetylgalactosamine. Now, if we look at the B type, okay, it's exactly the same as O, but it has this extra sugar here, which is called galactose. Galactose. All right. And the last one is AB. As you might have guessed, it's a combination of B and A together. Okay, so you can see this is the A, the blue one, and this is the B, which is the red one. So this is a very, very interesting example because it shows you how a very tiny difference, which is just one sugar unit, can actually lead to different blood groups. Okay, so the 4 human blood groups, they differ in the external carbohydrates that are bounded to the lipids. That's why we have the 4 groups of RBC, which is red blood cells.